Hello students, welcome back to our Physio-Pulse channels. Today, in the second part of the micturition process, we will discuss in a simple, easy to understand way and in an exam-oriented manner. Specially designed for you. And so let's get started. Do not waste our time. Let's get started our micturition second part. And actually, we have last class we have completed or the process, the functional actually how to fill up into a urinary bladder and what are the nerves which are involved in the urinary for the innervation process. And now to see one important thing that is the cystomatogram. It is a graphical record of the urinary urine volume fill up versus the pressures. And this is actually the different definition is systometry is a technique by which we study the relationship between the intravesical pressures and volume of urine in the bladder. So this is a graphical registration of the pressure changes in urinary bladder in relation to the volume of urine collected in it. How to record this? This is double lumen catheter is introduced into the urinary bladder, and one of the lumen is used to infuse fluid into the bladder, and other one is the used to record the pressure changes by connecting a suitable recording instrument. First, the bladder is emptied and completely, and then there is known quantity of the fluid introduced into the bladder, regular interval, and then intravesical pressure developed. The fluid recorded continuously. The graph is obtained by plotting the all values of the volume of the pressure, and the graph is called the systematogram (CMG). So this is described in the systematogram, and here you can see there are the four segment. Actually, segment one, segment two, segment three, and in this way, pressure and volume we can record it here. So if you have to describe this segment one, initially when the urinary bladder is empty, the intravesical pressure is zero. So segment one, when it is zero is the starting initial period. But when it reaches into the 100, then it completed the segment one, or the fluid is collected, the pressure rises sharply about 10 centimeters. So if you see 10 ml of fluid when entered, then pressure rises to 10 centimeter H2O. Now, in this way, segment 2, segment 3, we have noted here 300 to 400 ml in the segment, so it shows and remains 10 centimeter in H2O after introducing this. So it is because of adaptation of the urinary bladder by relaxation. So in accordance with the Laplace law. What is the Laplace law? There the P is equal to Q by R, where P is the pressure, Q is the tension, R is the radius. So according to this, the blood in the bladder, the tension increases as the urine is filled up. And at the same time, the radius is also increased due to the relaxation of the Detrusor Muscle. Because of this, the pressure does not change and plateau appears in the graph. So with 100 ml of urine and 10 centimeter H2 intravesical pressure, the desired micturition occurs. So desire of this micturiation is associated with the value of the filling in the perineum, but it can be controlled by voluntarily. So whenever an additional 200 to 300 ml urine can be collected in the bladder without much increasing the pressure. So however, when total volume rises beyond 400 ml, the pressure starts rising sharply. So in this way, you can say when urine micturiation should be happen, or what is the time exactly time when the micturiation or urine is ejected from the urinary bladder. Now, very important things is the micturition reflex. You see, the micturition reflex is a reflex of the by which the micturition occurs. That this reflex is elicited by the simultaneous stimulation of the stage receptors situated on the wall of the urinary bladder. That is the Detrusor muscles, which is carrying the stage receptors and urethra. Now, when 300 to 400 ml of urine is collected in the urinary bladder, then intravesical pressure increases, and this stretches the wall of the bladder, resulting in the stimulation of the stage receptors and generation of sensory pulses. So the pathway of this micturition reflex is given in here. So we are not describing in this micturition pathway, but importantly, I want to say that the higher center actually in the some important centers of the micturition is there, like the inhibitory center of micturition and facilitatory center of the micturition. This is the inhibitory center at the midbrain and the cerebral cortex that can inhibit the micturition by suppressing the spinal micturition center. But facilitating actually it can centers at once and facilitate the micturition via the spinal center, and some center of the cerebral cortex also facilitate the micturition. So these are the higher center for the micturition we are studying. But one important thing which we'll discuss now one important thing which we are discussing here, that is the how exactly how using this the process guarding or micturition reflex is working here. If you see this graph or this diagram, it shows that filling of the urinary bladder, it can stimulate the stage receptors. Now after stimulation of stage receptors, afferent impulses passes via the pelvic nerve. Then sacral segments of spinal cord, efferent impulses via the pelvic nerve, contraction of detrusor muscle, and the relaxation of internal sphincter, and flow of urine into the urethra, and also stimulation of the stage receptors, afferent impulses via the pelvic nerve, and inhibition of urinary nerve, relaxation of the external sphincters, and then voiding of the urine. In this way, urine is ejected from the urinary bladder. So this is all about the micturition reflexes and micturition process. And if we see the abnormalities of micturition, like there are so many types of abnormality we have found. One is the atonic bladder or effect of destruction of the sensory nerve fibers, like the atonic bladder is the urinary bladder which with the loss of tone, detrusor muscle, and so-called the passive neurogenic bladder or hypoactive neurogenic bladder. It is called also the destruction of sensory or pelvic nerve fibers up from the urinary bladder. So due to this destruction of sensory nerve fiber, the bladder is filled without any stretch signal to the spinal cord. So due to the absence of this stretch signal of the detrusor muscles, what it will do, it can specifically it can loses the tone and becomes a passive. So the bladder is completely filled with the urine without any micturations. So in this way, it is called sometimes it is called the overflow or in the drops as when the impact in the bladder. So it is called overflow of incontinence or overflow in the on dribbling. So the condition of destruction of sensory nerve fibers, that is spinally, if it is say a spinal injury, then it can be happen, or syphilis, then it also may happen. Another important thing is we have seen there is a nocturnal, very abnormality important. We have found nocturnal micturition. What is this thing? Nocturnal micturition is the involuntary voiding of the urine during the night. They are otherwise known by enuresis or also bedwetting, and it occurs due to the absence of voluntary control of micturition. So same thing, reflex is not working here. It is a common and normal process of in infants and children below the 3 years, and it is because of the incomplete myelination of the motor nerve fiber of the bladder. So when myelination is completed, complete and voluntary control of micturition develops and bedwetting stops. So even nocturnal micturition occurs after the 3 years of age, it is considered as abnormal. So it occurs due to the neurological disorder like the lumbosacral, that means lumbar and sacral vertebral defects, or it can also occur due to the psychological factors, loss of voluntary control of micturations, even during the impairment of the motor area of the cerebral cortex. So we complete second part of the lectures where we elaborately discusses CMG process and micturition reflex process, specifically the causes of micturition reflex or how it is occurred, and the important disease or disorders like that we have discussed here, nocturnal micturition process. Even also we discuss here like the atonic bladder as the effect of destruction of sensory nerve fiber. So students, we have completed our micturition total process, total chapters, and in which type of question where if you give some graduation or even also any other level of examination, if any questions comes, I think you can solve easily after doing this after reading these lectures or among these lectures. So so students, we have completed our second part of the lectures, and actually we have elaborately discussed here in the reflex system, micturition reflex process, and the exact mechanism of the micturition process. And also we have discussed here abnormalities of the micturations when the reflex is not working, uh, what are the abnormalities found, like the atonic bladder, effect of the destruction of sensory nerve fiber, even also uninhibited of the neurogenic bladder, even nocturnal micturition. And we also discuss here why the below 3 years of children and infants, their normal process of baby eating or also any basis is known. So In this way, we have completed our introduction and second part of the lectures. So students, if you have any queries, any questions, you can freely ask or freely write some comment, write in our comment box. And thank you for watching this lecture. Hope this session helped you better understand the topics clearly. And if you found this video useful, please like, share, and subscribe to our Physio-Pulse channels. Do not forget to press the notification bell so you never miss our upcoming lectures like the physiology, biology, and also biochemistry. So thank you for once again see you in our next lectures, and till then keep learning, stay curious,